This short video aims to illustrate the activities and results of the BeXyl project concerning the thermal treatments for the production of Xylella Fastidiosa-free nursery materials. But why do we speak of propagation materials? Xylella Fastidiosa lives only in the wood and is therefore transmitted and spread efficiently at long distances with vegetative plant propagation materials such as whole plants and parts of plants. Furthermore, due to its high genetic variability, Xylella can cause serious epidemic diseases in a huge number of important crops, ornamentals, and species of the natural flora. Today, it represents a major threat for the global plant trade and a true nightmare for European nurseries. The idea for a specific work package in BeXyl came from the previous Horizon projects, Ponte and XF actors. During their final meetings, nursery representatives explicitly required EU authorities to provide higher attention, research, and specific technical solutions to help the sector, which was affected by growing negative commercial impacts and damages. The proposed solution is represented by thermal treatments. This consists of the application of high or low temperatures using water, air, or vapor, capable of killing pests or pathogens outside or inside fruits and plant propagation materials. It is a physical, economically sustainable, and environmentally friendly system. However, it requires studies and validation of specific protocols and conditions of treatment, meaning exact times and temperatures, able to clean the nursery material without affecting its quality and vitality. Despite the existence of dated scientific literature and official standards for hot water treatment against some harmful organisms on a few host plants, like grapevine, pome fruits, and strawberry, this broad-spectrum phytosanitary tool remains largely underutilized. It requires further study to be applied against pathogens like Xylella and a higher number of important plant species. Besides nursery production, thermal treatments can find a wider application for the international exchange of materials for scientific uses and quarantine purposes. The research, conducted by a broad partnership including research institutions, nurseries, thermotherapy, equipment manufacturers, and 2 nursery associations, has been organized into 4 different tasks. First, to verify the efficacy of temperature and time combinations on the bacterium in vitro and in infected tissues. Second, to verify the effects on the quality and viability of plant materials without the bacterium. Third, to validate the best thermotherapy protocols on healthy material at a medium scale in nurseries. And fourth, to evaluate operational treatment conditions and alternative technical solutions. In vitro, using up to X strains of 3 different subspecies representative of the bacterium's genetic variability, as well as advanced fluorometric and molecular techniques, we evaluated the effect of X temperatures. These range from -15 up to 50 degrees Celsius, tested over different times, from a few minutes up to several days and weeks. We evaluated their impact on biofilm production, multiplication, and viability of the bacterium. Regarding the trials with hot air and hot water against the bacterium in vivo within the host tissues, we faced great difficulties with artificial infection. We used both potted almond plants, inoculated by microinjection, and cuttings of ornamental shrubs from naturally infected plants in the field. We successfully identified grapevine, naturally infected by the strain ST1, as the best model combination. Thermotherapy trials conducted with healthy plant material, whole plants for planting, dormant buds for grafting and cuttings, were carried out on a small and medium scale in laboratories and nurseries. We used different representative varieties of many host species: olive, grapevine, almond, cherry, peach, walnut, fig, white mulberry, oleander, lavanda, polygala, laurus, acer, elm, 5 quercus species, and others. As expected, although there are notable differences between species and varieties, dormant deciduous plants better tolerate heat treatments at high temperatures. For some other evergreen species like olive, even if the maximum conditions tolerated seems unable to eliminate Xylella, the treatment was verified useful to control other pathogens, such as Pseudomonas savastanoi. The olive and almond trees HW treated in 2023 were checked for microbiome modifications. Then, in 2025, planted in open field in Puglia for phenological observations and the detection of potential epigenetic modifications. To evaluate the viability and multiplication capacity of the treated plant materials, traditional techniques such as rooting percentage or grafting success rate were compared with new and faster methods. These include stereomicroscopic observation of the tissues, budding in water, and an electrical conductivity test called the Electrolyte Leakage Test. The trials, carried out mainly in Italy and Spain over 4 years, demonstrate that several important variables play a crucial role: the harvesting time, the level of dormancy, the diameter of the propagating material, its nutritional status, as well as pre- and post-treatment conditions. These must be further investigated with a larger panel of species and varieties. Similarly, some limited, very preliminary experiments with cold treatments and hot vapor could also be promising for future nursery applications. The most tangible result achieved within the project is certainly a draft EPO standard for the control of Xylella with hot water thermotherapy for grapevine, fig, and white mulberry. However, the large amount of new knowledge acquired through a huge number of experiments, including negative ones, requires continuation of the studies. This gives hope for expanding the standards to a greater number of species hosting Xylella and for using heat treatments against more target organisms. Finally, some important side activities confirm the interest and positive perspectives to extend this research line, ranking the nursery importance of Xylella host plants, classifying them by technical and physiological characteristics, the preliminary results of a questionnaire for nurseries, and the success of divulgative seminars, all demonstrate a strong interest in the future of these phytosanitary tools. Let's work together to get a sustainable and healthy future for plants and nurseries.